Yeah. So I researched the human neurodevelopmental disorder Rett syndrome. It actually shares a lot of similarities with autism spectrum disorders and actually used to be classified as one. It's an X-linked dominant disorder, so we see it primarily in females, and it's caused by mutations to a protein called MECP2 on the X chromosome. So MECP2 is methyl CpG binding protein 2. So it's a big epigenetic reader, but also really widely a transcriptional regulator. It's capable of both upregulating and downregulating based on where it binds. And the goal of my project is to really assess how mutations to MECP2 are contributing to changes in its binding activity. And for this, we're using a mouse model representing Rett syndrome, where we've essentially knocked out the Rett-relevant isoform of MECP2, MECP2E1, and we're comparing that to a wild-type mouse model across different time points to account for the progressive nature of the disease, as well as looking in very certain cell types. So we're taking a cell type specific approach to see if we can understand imbalances in the neuronal connectivity, so in different types of neurons. So yeah, that's my project.
That sounds incredible. But for people, for families specifically, they might not have the best understanding of even like genetics has to do with how their child is developing and how these factors would be important to their day-to-day life. Could you explain a little more in layman's terms how that's important and what the potential benefits of this type of study can be?
Yeah, so one of the biggest things about neurodevelopment is that there's very specific gene balances and transcription profiles, which is essentially you're expressing certain things at specific times so that the brain is developing properly. And when something goes wrong in that process, we tend to have downstream effects where you're not having proper development in the brain and you're starting to see that as physical manifestations. So this might be like you're not able to speak, or your language starts to go away. You're no longer able to walk the way you used to. And so it's a lot of loss of skills and changes in behavior that we see. And I guess the reason that we're doing this is to see if we're able to tie what's happening at that molecular level to the symptoms so that we can actually address that. So if we know what's going wrong in the brain, we can go back in and try to fix those things that are going wrong so that we prevent that cascade of events and we don't end up seeing those phenotypes or symptoms after all.
Thank you for that description. That's actually going to be super, super helpful as well. And another thing that I was wondering is, how did you decide on this particular factor to study? And how long did it take you to kind of get that replicated in mouse models and those types of things?
Yeah, so MECP2 was actually first characterized in the early 90s, I think just as an epigenetic reader. It was just a protein that's capable of binding methylated DNA regions. And it wasn't until, I want to say 1999 in Huda Zogby's lab where they tied that to Rett syndrome for the first time. And it's one of the few relatively considered monogenic disorders, meaning that only one gene mutation is responsible for the disorder. And we started with, in the field generally, mouse models where we've knocked out or truncated MECP2 and studied what happens with mutations. But MECP2 usually, when mutated in patients, is a point mutation, which is a single nucleotide changing to another nucleotide identity, and that affects the protein activity. So our lab actually recapitulated or recreated a mouse model based on a patient mutation where the start codon of the relevant isoform was changed from the start codon to a non-start codon, which means that there's no transcription happening of that isoform. So the senior scientist in our lab, Dagi Sui, actually created that model in, I think it was 2013 or 2014, and we've been using that model ever since. So that's where our focus is. And I think that it's a relevant model because this is actually the type of activity we see in real Rett patients. And on top of that, we're able to use both a male and female model, whereas the field does tend towards using the male model, even though female patients are what we see mostly.
Yeah, that's really interesting. And it's actually really great that you guys can have that more balanced replication, because I mean, that is one of the biggest problems in any scientific field at the moment, is that most of it has been done, especially in the medical field, just on a male model, or predominantly on a male model, I should say. So it's awesome that you guys get to work on that as well. How exactly did you kind of fall into this field? Like, what is your connection to it, and your little bit of your backstory?
Yeah, so when I was an undergrad, I actually also went to UC Davis for my undergrad. I took a genetics class with a professor, Alex Nord, and I was really interested in what he had to say and the research that he spoke about. So I ended up working for him during my undergrad, and his lab focuses on very broad, like autism spectrum disorders and other mental health disorders. And I found it really, really interesting. So starting grad school, I decided I wanted to stay in the general field of neurodevelopment and neurogenetics, which brought me to Dr. Janine LaSalle's lab, where we do Rett syndrome research and broadly other epigenetic and neurodevelopmental disorders. So that's how that transition happened. And I think since I was just interested in the broad aspect of neurodevelopment, I wasn't particularly picky about the disorder. And as soon as I started the project during my rotation, I absolutely loved it. So I continued.
That's awesome. And also, just by the way, for people that might not know a lot about Rett syndrome, could you give us like a small TLDR about it?
Yeah. So Rett syndrome is a human neurodevelopmental disorder that affects primarily females. It's considered a rare disorder, but affects roughly 1 in 10,000 female births. And the symptoms are very similar to ASD behaviorally. So we can see like anxiety, repetitive hand motions, but we also see more physical issues such as language and motor regression. So that's where people, or like children who are starting to learn how to speak and walk, lose those abilities. And this eventually goes to the point in many cases of immobility, not being able to communicate, being unable to feed themselves, and things like that. So it's a neurodevelopmental disorder that affects both behavioral and physical aspects of life.
Yeah. And so in early diagnoses, I don't know if you know anything about this either, because I know you're working kind of like more on the underlying biology of it, so you might not have as much of the diagnostic experience, but are there issues in diagnosing Rett syndrome versus ASD, or is like, especially in the younger years when the symptoms may be more similar?
Mm-hmm. I think there are. Part of the complication is that there is misdiagnosis as an ASD earlier on, and very similar to ASD, it's a little more delayed, so it's not really diagnosed at birth typically, and symptoms don't start showing up until about 6 to 18 months of age. There is more recent research, I believe, targeting early diagnosis mechanisms or tests, I suppose. But I think there are a lot of similarities in diagnosis processes and I think misdiagnosis. So I know originally a lot of these cases were classified, I want to say as an atypical ASD, and then reclassified as Rett syndrome. And a lot of times I know that that diagnosis flip-flops once more starts to develop.
Yeah. I guess the only way to really have been sure between the two of them would go through genetic testing, right?
Yes.
Yeah.
So this is just a thought that I had talked about with a faculty member at some point, but because Rett is largely monogenic, meaning we can like do PCR and amplify the MECP2 gene and sequence that, we could potentially have that on like the newborn screen or something. And I think that would be really nice to see, especially because if we are able to detect that so early on, early intervention is really helpful, like it is with ASD, in preventing severity of phenotype.
Yeah. Do you guys have any like preliminary findings at the moment, or like what are you seeing as a trend? If you can share, if not, completely understand as well.
Yeah, I think most of what we see so far are some of the metabolic phenotypes. So we've done some of the ChIP-seq or like binding profile analysis for some of the more wild type cells, which means they don't have the mutation, but it's telling us that normal MECP2 does play function in like the metabolism, and it actually plays a really large role in that. I know the lab published a paper on that in, I think, 2021, and we're building off of that work now. But other than that, we're still trying to get some mice to breed. So
classic, the glamorous life of science.
oh yes.
Oh yeah. But thank you so much for making some time.